Hello everyone, good afternoon and welcome to the 15th talk in the capstalks series. So today we are in the medicinal plants and a collaborative work theme, and our topic for today is the omics world of medicinal plants part 1. So over to you, Shailya.
Good afternoon, everyone. Thank you, Meenakshi, for the introduction. So, as we go into the world of medicinal plants, so we all know there are numerous medicinal plants around us and also in our kitchen, and which are being used in Ayurveda for a very long term. And so, we will be going deep into these omics world and see how we can scientifically know more about these medicinal plants. So to start with, we will be having three stories today. The first will be on Tulsi, which is also called as Ocimum tenuiflorum. The second story is on Perandai, which is Cissus quadrangularis. The third is on Methi, that is Trigonella foenum-graecum. So the first story today will be covered by Dr. Atul Upadhyay on Tulsi, where we will hear about different plant and leaf morphology of Tulsi and how there are differences between Krishna and Rama Tulsi. Also, he'll be showing some comparison about these Tulsi with respect to other plant genomes. Over to you, Dr. Atul.
Thank you, Shailya. Just confirm if I am audible.
Yes, you're audible. Please.
Okay, thank you. So let me share my slides. Right. Good afternoon, everyone. Thank you, Shailya, for the introduction of today's different stories which we are going to discuss. And the first part of this omics, plant omics, I am going to discuss about the genome and transcriptome data analysis of the Basil Tulsi. Right. So this was the first plant which we took to the genome and transcriptome level sequencing and try to find out the genes and their expression patterns related to secondary metabolite production. And then the first question comes, why Ocimum? As we all are aware that this is one of the traditional medicinal plant of Indian origin, and it's one of the holy plant of our country. So, and along with that, there are so many medicinal importance of this particular plant. I have listed few of them here, but the list is endless. So, for example, it is used in the anti-tuberculosis, antiseptic, anti-stress, anti-cancer properties, and many more. Right. And coming to about the botany of this plant, this plant belongs to the Lamiaceae family, which is also known as mint family, and the genus is Ocimum. There are 9 different species present, and if you look the chromosome number, that is the genome which we are interested in, the chromosome number varies from 2n equals to 36 to 2n equals to 72. So you can see that there is a huge variation in the genome. And with all these background, we started sequencing and analyzing the genome and transcriptome of the Tulsi plant, which I'm going to cover one by one. So first, we got the genome sequence in data in terms of paired in libraries and made paired in libraries. So when I say paired in, what it means, it's like small reads. When I say made paired, these are the bigger read sequences. So paired in are sequences which are sequenced in the opposite direction. The primers are designed in opposite direction, whereas in mate pair, the primers are in the same direction, and we get the bigger sequences. And it is important to have different libraries in the case of genome sequences, because the more the libraries, the better the coverage and the depth will be of the sequencing. So we got 2 libraries of the made paired and paired in. And the sequencing was done in the by Illumina HiSeq 1000 technology in the next generation genomic facility at CCMP, that is CCAM. And total we got 90 GB of paired in data and 26 GB of made paired data for Krishna Tulsi. So this is once you get the genomic data in form of reads, the first thing is to check the quality of the reads. If the reads are good quality, so that we can process further. If not, we need to do a lot of pre-processing before going to in the analysis. So first and most important thing what we check is the quality of these reads, and there are many different parameters on which we do. Most important parameter is the predisposed. So we can see that this is the, when I say R1 means the reads which came from the primer 1 and the R2 reads which came from the reverse primer. That means R1 and R2 are the complementary to each other. So R1, so quality of R1 was checked at Q20 P100. When I say Q20 means the Fred score 20 and P100 means 100% of the sequences. That means all the sequences should have quality of 20 or more. That was the criteria, and obviously we tried a different quality score and different parameters, and we selected this particular result for further analysis based on our different analysis. So as I told you, there are many different parameters which we consider, like GC content, percent of the reads which are discarded or retained, the adapters, and many more. So on the basis of that, we have selected this particular data set for further analysis. So we use this data for the assembly of the genome. You can see that this is the one of the results which says that genome assembly at Kmer 43 by probe de novo. Although we have tried 2-3 different assemblers, Kmer 43 is like K-tuple 43, which was the best result in this case. We have tried from a range of Kmer starting with 21 to till 63 at the alternate of at odd numbers. And then at the Kmer 43, we got the best result, and these are the results. You can see the scaffold, total number of scaffolds were 78,000, more than 78,000. So when I say scaffolds and contigs, I am sure that all are aware. Contigs are the assembled reads when you assemble the reads in the bigger sequences, and the scaffolds are when you fill the gap between the contigs. It becomes a more bigger sequences, larger sequences. So we look the assembly in terms of contigs and scaffolds. That means the number of sequences we obtain after assembling the reads. And N50 is the statistical value which tells you about the quality of the assembly. So the bigger the N50 value, the better the assembly. The one we can say in that it's a thumb rule. Okay, so and coming to the genome size, we have covered approximately 375 million base pairs, which is around 54% of the assembly of the genome size. One can say this is we are not covering a much, but since it's the genome is varying a lot of repeats are present in this particular genome, and there were limitation in the genomic library, so that at the first attempt it was good number. So once you have assembled the genome, that means you have a bigger sequences in hand. Now you want to find out the genes on that in these sequences. As we all are aware that the genic region are not not great in number as compared to the percentage of the genome. For example, in human genome, it's hardly 2%. Likewise, in most of the eukaryotes, there are a lot of repeats and non-coding regions. So we have to find the coding region, that is gene, and then annotate that gene, what that gene is actually, that sequence, what it is going to code for, which protein. That comes under the annotation. So that was performed by using this particular pipeline, and we in the first attempt, we got around 36,000 genes with average size of 2,400 base pairs, and total length of these genic region is 86 MB. And this was the 23% of the genome. You can see that percentage of the genome covered here is very high because a lot of the repeats were not sequenced or not assembled in our case. Okay, coming to the pipeline we use for the annotation. This is one of the standard pipeline. So we provide our input sequences in terms of, you can say, mate, paired, and paired end, which are assembled, and then this will become our input sequence. Then there are different stages, like we go for the masking of the repeats, identification of the repeats by de novo approach, or maybe by already known repeats, and mask them. So this step is very important because otherwise it will hinder in the gene prediction. Likewise, we have used all these different tools, and finally we annotated the number of genes. Coming to the next slide. So once we have the genic information, we also got the transcriptome data of these two cultivars for different samples. So what we did, as the genome was not very high quality, like in terms of coverage. So what we did, we tried to assemble all the transcriptomic data together and make it as a reference. And then by using that reference, we have tried to find out the differential expression of these genes. So the pipeline was something like we assembled all the transcriptomic data and then got NR by using CT hit at 90% sequence identity. And then we use that NR combined transcriptome as a reference, and we use Krishna and Rama samples separately to map to this particular reference and then identify the RPKM values of all these genes. And then along with that, we also filtered out the genes which are involved in the secondary metabolite pathway by using the genomic and approach. And then we looked the expression of these genes separately by using this transcriptomic data. So coming to the results of the RNA assembly, as I told you, this is the combined assembly results, and then we also have the separate assembly of each samples. I'm not going to read all results, but this is the combined assembly which can give you a insight like how we got the results in form. Like again, assembly, number of contigs and scaffolds. We also looked the N50 size and the percent of GC content, and all were checked, and then this was used as a reference. So by using that as a reference, we mapped all the reads from different samples at different stages to the reference and find the differential expression. And here you can see the heat map of the top 50 transcripts which were differentially expressed in these two samples. There were more than 200 differentially expressed genes, but we have plotted only top 50. And most of these top 50 are at least 8 times regulated in terms of down or up regulated. Then we look another part of the annotation is like you try to find out the function in terms of domains it contains, the domain architecture, and many more different terms. So first was looking the number of domains which were present. So basically from here our analysis was constrained only to the metabolite producing genes. So this data says that these are the domains which are more prominent or present in high number in the metabolite producing genes. Most of them are the kinases, different type of kinases, and then P450 and many more. Likewise, we also try to look the orthology of these secondary metabolite producing genes in different plant genomes. So we have selected 4 of these genomes, and then we found that there is a huge similarity in terms of metabolite producing genes among these plants, which also indicates it's not a random drift. It's a continuous evolutionary process. Some of the secondary metabolites which we were interested, or these are the mainly present metabolites in the Tulsi. Some of them which we are interested, and we further go ahead and to do the RT-PCR, qRT-PCR. The examples like eugenol, oxalic acid were used for. So qRT-PCR is like we want to further validate for few of our results, like transcriptomic data itself tells us about the expression of these genes, but we want, since transcriptomic is the omics approach where we have used we check the global expression, there are chances of the noises. So to confirm if our data is correct or we are on the same line, we also try to do qRT-PCR, and we selected few of these genes and we did the analysis, and then we found that there is correlation between the transcriptomic data and the qRT-PCR results. With this, I think I have covered most of the things. Since we have done a lot of work here, but because of the time constraint, I am going to stop myself here. Thank you everyone.
Thank you, Dr. Atul. So as this was the first project venturing into medicinal plant, ma'am to say a few words on this initiative.
Okay, yeah, this project in fact is the first amongst the plant genomics transcriptomics works that our lab started pursuing, and this is actually a drift, as it were, to move from plant stress management to actually going into the plant genomics areas. And I will talk about it a little more towards the end of the seminar. But coming back, this is the first project amongst the plant genomics ones. And definitely it was self-motivated, but in a way, this drift was the most unexpected as well. However, as you will see, this has led to very exciting and a rewarding experience. Thank you.
Thank you, ma'am, for sharing those insights. So now we will move to the second talk for the day. So the second talk will be by Dr. A. Methi on genome and transcriptome analysis of Cissus quadrangularis. So this plant is well known for use in medicinal purpose as analgesic, anabolic, and so many properties. So over to you, Dr. Methi.
Hello, hi all. Thank you for the opportunity. So I'm really happy to, look back at the research, what all we worked on, and just reminded about the time where the whole the group members were discussed on the genome. What are the tools and what are the best approaches for genomic research, especially on the plant genomes. So I've just kind of reminded of with those moments. So thank you for this opportunity. So let me share my screen. As Prof. Sowdhamini mentioned that, that was the first experience working on Tulsi for all of us. Then moving forward, we all got so much excited about, especially on the medicinal plant. Like, why don't we focus on other very important medicinal plant? Can we also, take up such important medicinal plant and do a deep dive into it and find out what kind of an important secondary metabolites and what are the pathways which are involved in producing those metabolites. So that's in that line. So there were multiple plants were taken, and one of them is an important plant called Cissus quadrangularis. I do not have much details to present, but the reason I just wanted to give a brief about how we started with this. So this is a plant, I think, I don't know how many of you have seen it, and this is a creeper plant, and it prefers dry land for growing. And this plant has been used in traditional and classical medicine in I don't know, in South India especially, for improving bone health, right, and also weight management. So so the metabolites, secondary metabolites present in this plant are really having a very good medicinal values. Especially you can see this plant where it's a lot of stem, and it has very less leaves, but the stem is the most important, and it carries a lot of medicinal values. So that's what the introduction all about. And definitely we the reason why we took this plant is to do genome sequencing and transcriptome sequencing, and to decipher whatever the secondary metabolites and the related pathways. As you have seen, these plants are very unique, right? It's not like something it is well known, well established or well studied plants. So we also had difficulty in looking into the metabolomic pathways, like what are the metabolomic pathways which are well studied, and can we able to understand the pathway information, or what are the enzymes involved in. So what we decided first, we wanted to just work on the genome. So moving forward, I'll also talk about phytochemicals. What are the important phytochemicals present in this particular plant? So as you can see, it is mainly used for fracture healing, and it is used for weight management. And these are the important secondary metabolites, which are quercetin and camphorol, are very important stilbene derivatives. And there are other resveratrol and other metabolites also in, which has a very good medicinal implication. So so this is like something we wanted to do a study of what is present in this plant and why it is so much value it has, right? So there is a review paper, and this I also wanted to tell you something. In Tamil Nadu, this plant is being consumed in a form of a chutney, right? And it actually gives a lot of good health in terms of the bone, if there is a bone fracture or weight management purposes. So even now it is being consumed, and it is traditionally followed. Classical medicine, basically, if they follow it, they consume it. So it has a lot of calcium, and it's actually rich in calcium ions, right? So that's also reflected in this paper, that what are the important medicinal values this particular plant has. So when we started this sequencing, so as you have seen in the introduction, this particular plant belongs to Vitaceae family. basically, it has a very close relationship with this grape plant. So when we started, we found, okay, what is the genome size of a grape, and we found it was 478. And in that, because definitely same family, they might have a similar, like, the similar range of genome size. So we went ahead for around 90x of genomics coverage, right? Sequencing coverage. So total number of reads what we have generated for the genome is around 147 million reads in the paired end. So as Atul mentioned, what is paired end and mate pair? So here for the genome, we have sequenced paired end data. And recently in 2019, there is a paper and they estimated the genome size, which is around 689 MB of haploid genome. So with that calculation, I think what we have is 64x coverage of the sequencing depth we had. And we use these reads for, whatever the steps we have followed in Tulsi, because that Tulsi was really a comprehensive learning from all the aspects in terms of looking into the tools, in terms of best practices for the genome, and also looking into the downstream consumption or downstream analysis of the genome. So that has given us a lot of experience. So with that experience, we went ahead and assembled the genome. And same way for we have also done RNA sequencing. So as I have shown you, leaf and stem has a lot of good medicinal values, and that has been published, well studied as well. So we have sequenced stem with 2 replicates. So RNA-seq sequencing usually done with replicates to basically, to have a very good confidence on the expression data. So we had around 23 to 39 million reads for the stem and 27 to 30 million reads for the leaf. So we have also assembled RNA sequencing data. So for the first round, what we have done is we wanted to announce to the scientific community that we are working on the genome, and we have assembled it. So only the genome part we assembled, and we did a step called, because as you know, plant genomes, there's a lot of repeats. So we definitely have to, check the repeats. So what is the percentage of repeats and things like that when the assembly is done. So we have followed certain steps like we did in Tulsi. We have done the genome assembly using soap de novo, then followed by repeat masking. And then we have taken those genome further for, maker annotation and downstream analysis. So this paper, which was published in 2016, we have focused only on the assembly and the annotation. So we could able to assemble around 312 MB of the genome with the total number of scaffolds of around 336,000, right? And the number of contexts and GC. So this is like a basic statistics any genome paper will give you. So this is the a very short communication on the genome part. So then we have also worked on the transcriptomics. I think there are still some work needs to be done in terms of, looking into the metabolites path, because it's just an assembly. I feel the current team members are working on it. So the transcriptome assembly, as you have seen, we have done assembly separately for the stem, leaf, and also combined assembly. The total number of transcripts what we have identified is approximately around 60,000. And obviously, the transcript might have a different length. Sometimes they might be even small as 30 bases, or even it can be bigger size also. So we need to segregate them, whether we are really looking into the CDS region or the transcript region. So in that we have done certain filtering and looked into the N50 values. So this is a very preliminary result what I'm showing here. In the interest of time, I'm not going into details about pathways and other things. Yeah, so this whole thing got published in 2016, and more work had to come. And these are the members, like, from Tulsi, basically I think all the members of CAPS who worked on this plant genomics initiative. And I think that's where I got so much inspiration about moving into the genomics field. So yeah, thank you so much, ma'am, for the opportunity. And thank you all for listening to it. Thank you.
Thank you, Dr. Methi. That was really nice. So we'll move to the third talk now. So this will be by Dr. Mahantesha, who will talk on the transcriptome of fenugreek, also called as Methi, and the genes involved in the biosynthesis of medicinally important metabolites. So the Methi plant has a lot of different components like flavonoids, volatiles, alkaloids, and so on. So apart from that, we will also discuss some of the transcriptome-based results where different differentially expressed genes from leaves, stems, and roots, and also some of the biosynthesis pathway. Over to you, Dr. Mahantesha.
Shalina, thank you very much for your introduction and introduction of the crop. So first, I would like to thank Professor Sowdhamini ma'am for giving me an excellent opportunity to connect with the Lab 25 from 2009 as a collaborative PhD scholar from US Bangalore. That is the time I started coming to the lab, and then 2010 I actually joined the lab for research, and that's where my real learning of scientific research started with ma'am, and I must thanks to ma'am for her excellent guidance. And after that, so we were working on the mainly stress component, mainly for plant stress biology component, and connected with Dr. Shamir and again Dr. Uman and other lab members. Yes, of course, as we started working on that again, it was a wonderful opportunity for all of us in the lab where ma'am initiated about the omics approaches in the plant biology, and we all are very, like so much interested to learn, and so many things was absolutely new to us, and it is because of ma'am's support and encouragement and the team efforts of all the lab members. After working with that, we got an opportunity to work with some of the medicinal plants. So now what we did is, as a group members, we selected fenugreek as one of the very important plant. Yeah, so I'll be talking about a few aspects of the studies what we have conducted in the lab. So why we have chosen fenugreek? Because as you some of might be knowing that actually this fenugreek word is very common to all, and you are using this crop in your day-to-day life. And here, when you look into the crop itself, in fact, it is a very old medicinal plant. And another very interesting thing is, again, this is the originated from again India and Mediterranean region. A very important thing for this plant, though it's one of the very important medicinal plant, if you look into its cultivation, because this is a medicinal plant, but its cultivation in India, Nepal, Pakistan, Bangladesh, Turkey, some part of the world, because considering its importance with respect to the many aspects. If you look into the species richness, it has 140 species. So out of which, it is like 107 species which occurs in Asia only. When it comes into India, so mainly we are growing two species. One is common Methi, it is generally known as Trigonella foenum-graecum, and another one is Kasuri Methi. So that is again Trigonella foenum corniculata. So these are the two species we are commonly cultivating in India. Here, this medicinal plant is considered to be a multi-purpose crop. Why it is considered to be a multi-purpose crop? Because of its usage as a herb, as a spice, as a fodder, as a nutraceutical compounds, and nutritional constants. So having into such a rich usage of this crop in terms of multi-purpose usage, so that's why though the crop is very small, but if you look into the importance for the usage, it having multiple usages. For that reason, it is considered to be a multi-purpose crop. Having multi-purpose crop, again, very important thing of this crop is from US FDA, they have given this crop as a generally recognized as safe. It means that if somebody is interested to develop any of the compounds or any of the food products or any of the derivatives of this particular plant, generally they don't have any toxic effect on their health. So that is the one of the very important thing one can learn from this grass. So having into all this, and of course, especially for agronomical practices, this crop is given a lot of importance for the production aspect. But in terms of its medicinal as well as in terms of its molecular aspects, very less study is done. So looking into those aspects, we have initiated this work. And looking into its traditional usage of this crop, if you look into the usage of this crop, I told that it is one of the oldest medicinal plant. Again, that too from Fabaceae, Fabaceae family. Now, if you look into the history, so ancient Egypt people, they used to use this particular plant products for age childbirth, then increase milk flow. Now, again in modern Egyptian women, what they are using it relieving menstrual cramps, again making Hilbert tea. So again, this Hilbert tea is, in fact, it has its own wonderful story. So please do look into the Hilbert tea, why they prefer this. Taking this tea, it has a lot of medicinal properties. And again, it is using for baking bread. So having into this, so again, if you look into the Chinese people, so they are mainly, it is using for treating abdominal pain. Of course, as you know that in India, so it is very common food product is called as fresh methi ka saag. Again, many people, they use it in paratha. And of course, if you take off any of the dishes, it is mainly using in flavoring agent. And again, in India, Pakistan, Bangladesh, and again, some of the Asian countries, this remains as a very important popular crop, especially it used in the curry powders, again, putting into the pickles, and also it's one of the very important ingredient for spice mixture. And again, in Ethiopia, it is mainly used for baking bread. And still it is in European countries, they are using centuries as a cooking spice. US, yeah, they are using as a spice blended for the soup and stews. Again, in Swiss, they are mainly using for as a flavoring cheese. So by looking into the its traditional usage, a very important thing, it is considered to be a folk medicine for treatment of cellulitis, then boils and tuberculosis. So these are the very common skin diseases, and tuberculosis, TB, it is again very important disease where this particular plant is used. And in addition to that, traditionally, when you look into the traditional usage, so seeds particularly, they are using as a orally, particularly as you know that insulin substitute for the reduction in blood glucose. Having its total traditional usage, this crop got lot of interest and opportunities to look into its medicinal properties in terms of a nutraceutical aspect. If you look into the importance of this particular crop, so it has got a wide scope and opportunity to look for its anti-diabetic property. And again, it has in the anti-hyperlipidemic, anti-obesity. Again, if you see that there are a good number of anti properties it is having. Now, when you look into the plant, what we have selected, and further its omics approach, especially for transcriptomic approach, to look into the downstream of the analysis in order to identify a very important secondary metabolite biosynthesis genes. So initially, yes, we have selected the fenugreek plant. So from that leaf, stem, and root samples were taken, RNA is extracted, and the quality of RNA was looked into it. And later, using the HiSeq platform, sequencing was done, and the obtained raw read. So here, especially for the bioinformatics workflow, thanks to Dr. Atul and Dr. Methi, in fact, they had discussed about how the reads were obtained and how these R1 and R2 reads were assembled, and how further downstream analysis usually done. So having into the raw reads, once you obtain the high quality reads, a de novo transcriptome assembly was performed, and here all the three transcripts were pooled, and later that CDS were predicted, and from that, again, functional annotation were predicted, domains were looked into that, and further signal peptides were predicted, transmembrane helices were predicted, RNA sequences were predicted, and in order to look into the very important metabolites with respect to their pathways, so KEGG pathways analysis was performed. And again, in order to look into the similar genes in the other species, and using ortho-MCL tool, orthology analysis was performed. And ultimately, in the tissues, how these genes they behaves, in terms of their upregulation and downregulation. So using Express and RCM tool, so differential gene expression were carried out, and so they identified some of the very promising molecules. Further, they taken for the real-time validation in order to look into the tissue level expression. So the qRT-PCR is used, and again the candidate genes were validated. And again, it was quite interested to look into these metabolites, their abundance in terms of the tissues. So tissues were selected, and their secondary metabolites were quantified. So this is the brief about the total outcome of the assembly. So looking into the total transcripts, this is the total number of transcripts. Again within that, when we look into the the transcript which are more than 1 kbs. So nearly you have 48,000 of more than 48,000 of gene transcripts were identified with N50 of 13A2 and having the size of 169. And looking into the total GC content in the transcript obtained, it is 36%. And so again, having to the blast text, these are the again data which represent about the outcome of the assembly. And some of these are the total open reading frames are predicted. And again, further they are utilized for the, as I told that in the methodology, some of the downstream analysis. And these are the outcome of those predictions of the protein hits domains and signal peptides. So likewise, the orthologous groups. And this, the obtained, the generated raw data, it is submitted to the NCBS SRA, and this is the ID. One can look into it and see the raw data of the whole transcriptome. The main important thing to look into the identification of the genes which are involved in the biosynthetic of secondary metabolites. So here in-house protocol was developed, like so in order to identify the functionally important residues. So we have adopted the in-house developed protocol, so which is a knowledge-driven protocol for the prediction of functionality of the selected metabolites. So here what we have done is like mainly looked into the alkaloids. Again, in the alkaloids, trigonella and glycine butane, because again trigonella is one of the very important component with respect to the medicinal point of view. Again, glycine butane, in fact, in the plant biology, it is one of the very important molecule which is very much helpful for drought tolerance. So looking into that angle, we have worked upon it. And of course, yes, saponin, that too particularly a diazinin, it's a very important sterol molecule in the fenugreek, which is again having a lot of importance with respect to the medicinal point of view. We also looked into some of the volatiles, especially Medicinal and linol. So here, because these are the flavoring agents, and also looked into some of the flavonoids, very interesting, some of the flavonoids also looked into it. Here, looking into the trigonella pathway, so how exactly this trigonella synthesis is. So initially it's synthesized from the pyridine nucleotide cycling, so from that nicotinate form, and further again by the methylation, so it will form the methyl nicotinate, and again this is the trigonella compound. So here the enzyme is nicotinic N-methyl transferase, which will play a very important role for your synthesis of trigonella. Here the interesting thing, look into its functional important residues. So here if you look into and this is the transcript which is functionally obtained, which is having the character, like having the trigonella gene functionality. So here if you see that there are 3 substrate binding motifs were identified, and again one of the catalytic binding motif is identified, and again one of the SAM binding motif is identified. So this is very quite interesting outcome we got, and again this is this kind of identification is possible through again in-house developed a tool within guidance with Professor Sowdhamini ma'am. Now having into that particular gene, like looking into this trigonal gene across the different species, so multiple sequence alignment is performed, and when you look into the phylogenetic tree here, identified transcript, it is gave a very close relationship with the Medicago trunculata. Again, it is having the same tribe and having into 92% of identity and 100% coverage. So, and yes, of course, now based on this molecule is further validated using the real-time PCR in order to look into its like expression level in selected different tissues. And very interesting thing found here is we have noticed this particular gene, it gave its high expression in leaf, followed by stem and root. So here clearly it shows that the transcript what it generated through the transcriptome approach. So we have identified that. We have also looked into its functional residues in it, and also validated using the real-time PCR. So that's again reflects the quality of the transcriptome data generated and the gene identified and validated. So the second very important molecule is again diazinin. So this, if you look into here, this is the pathway of diazinin synthesis. It starts with the cholesterol. Interestingly, in our study, we have found very important enzymes. One is sterol 3b glycotransferase and another enzyme is beta glucosidase. So these two enzymes transcripts were identified. Further, looking into those transcripts functionality, the same protocol is followed. So, and having into that, this particular enzyme, it has two domain motifs. One is putative steroid binding domain, and another is the plant secondary product glycotransferase motif. And this, if you look into here, this particular enzyme, again, it got the two hits. And again, interestingly, this enzyme is also very closely associated with the Medicago trunculata, which is the very similar species. And next, another very interesting enzyme. So in that also, the similar line of similar protocol is followed, and we looked into their interesting binding motifs here. So in the same, again, enzyme, two motifs were identified. One is IV10 motif, another is the TFNP motif. And same thing also here, the transcript which is having this particular enzyme, and again looked into the similarity with the other crop species. In order to look into this transcripts obtained through NGS, some of the very interesting genes were identified based on their TPM values, which are having a good number of reads. Now, if you look into the different tissues, like particularly in leaf, stem, and root, so these are the very interesting genes which are having high relative expression of omics. And in stem, again, a very interesting gene it is given is the PR protein, means pathogen response protein. And these are the, again, when you look into the root, in fact, root it is melatonin and dihedrin. So these are the very promising genes, especially for plant breeders who are working or targeting for development of the either biotic stress tolerance plant or else even abiotic stress tolerance plants. So these are the candidate genes which could be effectively utilized for the crop improvement program. And another interesting thing, what it is noticed in the DEG, differential expression genes information, is the 6 transcripts, particularly these 6 transcripts. So they were shown high abundance in all the 3 tissues what it is selected. And in order to look into the quantity of this particular secondary metabolites, so since the targeted metabolites were like diazinin, trigonellin, and 4-hydroxyleucine. So since these are the 3 secondary metabolites which are having the promising compound for the development for any of the medicines, in particularly for curing some of the diseases or managing some of the diseases in humans. So if you look into the diazinin, especially in the seed, so if you look into the seeds as such, so highest this particular compound, if you look into the stephanoin, it is found 1.56 microgram per milligram of tissue what it is taken, and in the root it is 1 microgram per milligram. And if you look into the 4-hydroxyisoleucine, so it is found 84.77 microgram of milligram. Like if you look into the quantity of the this particular compound in the tissues, and again in the trigonellin, so if you look into that leaf, it is found 4.71, and again in the seed it is so 2.6. What it indicates is that the trigonellin compound, what we noticed, in fact, in this particular variety, it is found more in the leaf compared to the seed. So these are the very interesting things which came from the metabolomic quantification study. So with this, yes, of course, I would like to thank ma'am for giving us an excellent opportunity to get into the again era of next generation sequencing and get into the learning. It was absolutely a lot of learning experience with the ma'am and the team members. So I would like to thank ma'am and all the team members, and definitely with the previous lab members who have helped a lot in the discussions and in the helping for the coordinating the work and working together and come up with some good outcomes. So once again, I would like to thank ma'am for the given excellent opportunity to work with the team. Thank you, ma'am, and thank you all.
Thank you, Dr. Mahantesha. I would now request Shafi to please present the publications relevant to the talks.
Hello, everyone. So these are some of the relevant publications from today's talk. So first we heard from Atul about Tulsi genome, and he talked about the assembly and analysis of genome, and also he added transcriptome sequencing and analysis of Krishna and Rama Tulsi types. And next, we heard from Methi about Perandai genome and also transcriptome assembly and analysis. And this is one announcement paper of genome, what we have given here. And third, Mahendra talked about fenugreek transcriptome, and he explained the transcriptome assembly analysis. Following to that, some relevant metabolites and its analysis. So for next week in Caps Talk, we have part 2 of the Omics world of medicinal plants, and we will be mostly talking about drumstick plants and shangupstri plants. So please stay tuned for the next.
Thank you, Shafi. I would request ma'am to share a few more words about how these projects went ahead and what were the idea behind this. Over to you, ma'am.
Yeah, great. Yeah, thanks for the very smooth coordination, Shaya. And thanks to the speakers, Atul, Methi, and Mahendra for presenting the 3 works very clearly. Yes, so just to add a few words, definitely our interest towards genomics of medicinal plants was initiated because of our quest for looking for more functional variety in enzymes and the enzyme superfamilies, and that's why we started picking up medicinal plants. And there are also some kind of impactful experiences that led me to this area. And I must say that we had shortlisted about 25 different important medicinal plants. And for a while, this whole drift from plant stress management to plant genomics was kept as a closely guarded secret. And I was sharing it with Professor Ramaswamy, who was then a colleague in STEM, simply because he was looking after the sequencing facility and I was looking after the computational arm of the sequencing facility. And as this was going on, he also mentioned that he happens to have, that he meaning Dr. Malali Gowda, our other collaborator, and him, they happened to have raw reads of Tulsi, and we definitely jumped on that idea. And this is how, the projects started in a self-motivated way, and then the collaboration began. Other thing is, when I decided to then work actually on the Tulsi genomics and transcriptomics, I asked in the lab who are all interested in to participate, and then literally everyone had raised their hands. They all wanted to be volunteers. And the beauty is just none of us had ever worked on this, and it turned out to be a very nice experience because we were learning and performing at the same time. And we decided to put our own self-driven deadlines, and after a while we were kind of meeting every day and talking about it, and it was a nice experience. But what we had not realized was this is going to give rise to a nice future career for most of them, because whereas the members, many of the members have now left the lab who had worked on these 3 projects. They are happily absorbed either as a postdoctoral fellow or a faculty or head of the bioinformatics teams in companies, basically focusing on genomics and transcriptomics.
So thank you, ma'am. Thank you for sharing all those old stories, which we won't be getting to hear otherwise. Thank you so much. And thank you for giving me the opportunity to host the event today. And also thank you to all the speakers. So first talk was by Dr. Atul Upadhyay, who is currently an assistant professor at Department of Biotechnology, Thapar Institute of Engineering and Technology, Patiala, Punjab. The second talk on Perandai was by Dr. A. Methi, who is currently a customer success manager at Elucidata, Bangalore, Karnataka. And the third talk on Methi was by Dr. Mahendra, who is currently an assistant professor at UHS, Bagalkot, Karnataka. Thank you everyone for joining. Stay tuned and see you next week.